Hello. Welcome to this FLASH mechanism session. It's a pleasure for us to introduce you to Joao Seco from the German Cancer Research Center in Germany. And he's going to talk about the physico-chemistry after flash irradiation and oxygen contribution.
All right. Thank you very much for the invitation to be here. And I want to thank everyone to be here in presence. I think for the online, I have to switch on the pointer. Okay. So I have no disclosures, and I'll move on. So the talk that I'm going to give is going to be divided into two main areas. The first one, I'll be discussing how membrane structure and composition can affect how oxygen diffuses through the cell and potentially create hypoxic nucleuses. And this has a big effect on things like OER and, of course, on the flash. The second part, I will discuss then the physico-chemistry and how potentially scavengers can affect the amount of reactive oxygen species present and how this is differential from healthy to tumor, of course. All right, so let's start with a simple concept of oxygen. This is a very well-known extracellular concept, and I will discuss also the intracellular. The extracellular, you know very well from the books like Eric Hall's book and stuff like that, that you have a specific distance from any blood vessel where you'll have potentially an hypoxic cell. So the distance can vary between 70 micrometers to sometimes 100, 120. And this is basically driven by microenvironment effect. What I'm going to mention now is the intracellular diffusion. So how is the oxygen diffusing in the cell, and can it be affected by properties from the membrane? And this is the key to something we published a few months back. So oxygen spatial distribution at the intracellular level, this is what I'm going to focus on this first part. So the current paradigm when we look at oxygen studies is that we assume that the oxygen is diffusing from the outside all the way through all these organelles, and eventually a certain amount of it reaches the nucleus. And there we have then eventually the DNA damage in the case of radiation. And what we make an assumption is usually that oxygen outside the cell, the extracellular matrix, is the same in the cytosol. It is the same in the mitochondria, it is the same in the ER and the Golgi apparatus, and eventually it is the same in the nucleus. So basically the whole surrounding environment works as a reservoir of oxygen. It's like when we are breathing, we have the same situation. The environment around us, the air, is loaded with oxygen, and when we grab the oxygen from the air, this oxygen is then replenished very quickly through diffusion. And so we assume that in these cases, the diffusion is much more efficient or faster than the consumption. So this is what we do when we do standard oxygen studies, that to the point that we don't even think about which oxygen is the one we're looking at. We just say oxygen studies. And so we always know that the extracellular matrix equals the nucleus. Okay? And this is an assumption that we make implicitly when we do the oxygen studies. And I want to raise the question that maybe this is not the best way to go, that there is a possibility of the oxygen diffusion changing through the cell. So I've just called it the new, but it might be a controversial comment, so I'll just leave it as the new possible paradigm. And in the case, imagine that the cell has the ability to control both diffusion and consumption, that it can reduce the diffusion of oxygen going through the cell, but it can also amplify the way it consumes oxygen, so make it faster, right? So you have these two things, and if this is possible, then the cell itself can alter this paradigm, right? Making the consumption stronger than the diffusion, and under these conditions you'll have a gradient of oxygen through the cell, and potentially you'll have low oxygen in the nucleus, potentially even reach levels of the hypoxia. So in these cases you have this gradient, and eventually you have an oxygen in the nucleus that is much lower than the extracellular matrix. So you have this type of condition, and we showed in our study that this is really dependent on potentially the cholesterol level that's in the membrane. So on the left side, you see in blue, here you have your normal phospholipid membrane, bilayer phospholipid, and usually these phospholipids work as a fluid system. They are very flexible, and in the transfer through the oxygen through it, I will show, is really efficient, very fast. Once you add cholesterol to the membrane, you basically make the whole membrane rigid. And it's much harder than to allow oxygen transport through it. And this is an extreme case with no cholesterol and cholesterol. But in reality, most of the membranes have a mix of these two things. And I'll show why cholesterol is important to the membrane. So here's an example of your phospholipid. You see the polar head of your two phospholipids, and you also see the polar head of your cholesterol, which is the little red dot. And you see the four rings, the steroids. sterols. And you see that in this region where you have the 4 rings, it is extremely stiffened. The whole region forces the phospholipids to stop moving. And this is how the cholesterol works. It basically enforces a rigid structure around it. And basically in regions where you have cholesterol, you have a much weaker diffusion of oxygen. So he has an example of one, and there's others over here. So the cholesterol is basically stiffening the membrane in the proximity of where the cholesterol is connected. And why is that important? Well, cholesterol helps with the temperature dependence of the membrane. So the membrane has a certain temperature dependence, and if it's extremely rigid or if it's fluid, this is really temperature dependent, and there is a transition temperature called the TM. And with cholesterol, this transition temperature becomes much smoother. If you have no cholesterol, then you have a very fast transition of the membrane structure from a solid-like to a fluid-like. This is heavily dependent on the chain. How long is my phospholipid? So the smaller phospholipids with 14 carbons, they have a very low temperature transition, while as you get longer and longer, you tend to have a stronger, a higher temperature transition. And most of the human cells have around 14 to 22 carbon atoms. So you see, we sit right in the middle of this region where we're very strongly temperature dependent. So what is the lipid content, the cholesterol content at the different areas of the cell, right? So the most important is the extracellular matrix, the plasma membrane. Here you have a very high concentration of cholesterol, and that's the number basically that we use. And the PL represents the phospholipids, and the chol represents cholesterol. And you see a very high concentration of the phospholipid membrane outside. Then you have different levels of concentrations depending which organelle. So here you have the ER with relatively lowish, but still enough. Then you have also the mitochondria. The endosomes have also extremely high level of cholesterol to phospholipids. And then you have others here. What is not shown is the nuclear membrane that also has a pretty high level of cholesterol to phospholipid. So you see that this concentration of cholesterol in the membrane varies significantly in the cell, and it has a very high impact in the way oxygen will diffuse in principle through it. So let's look at the transport mechanism. So there's been studies done over many years in the group from Subzinski, and he studied different levels of cholesterol in the phospholipids. He has an example of a phospholipid membrane without cholesterol, and on the vertical axis is the time of the oxygen to transfer through the medium. And here you have a barrier of water, and you have your phospholipid, and you have the temperature dependence on the vertical. And you see that through water, the time to cross water is slower than crossing a phospholipid membrane. So in this case, when there is no cholesterol, the oxygen transport through the phospholipid membrane is extremely fast. But as soon as you add cholesterol, you add a barrier to the oxygen. So you have a sudden increase as an oxygen barrier. So cholesterol is literally blocking this range of transport. And then, of course, there's the interbarrier, right, this is midway through the membrane, where it's highly efficient, right? So you have a barrier on both sides that blocks oxygen transport through the membrane. And another follow-up study is also from that group, published by Rachel Dodson. They even studied the oxygen permeability in the form of a velocity. And you see that the cholesterol content basically lowers the velocity oxygen can cross through. And the differences are small. They only order 5 to 10%. But that's just one membrane. If you think about the fact that there are dozens of membranes from the extracellular matrix to the nucleus, you see that 5% times 20 or 30 membranes has a huge impact. So what we did is we started to study this as a diffusion problem, and we added the So we studied this as a diffusion problem. And so we had the problem of studying how oxygen would diffuse through all these membranes all the way reaching to the nucleus. So on the right here, you have the extracellular matrix. You have all these different membranes. You have the ER and Golgi apparatus, the cytosol, and the nucleus. So it becomes a diffusion problem with a consumption function. The consumption follows a Michaelis-Menten type consumption, which is pretty common in the cell. And we modeled this in different regions slightly differently because the consumption has to change depending on the region. So here's the consumption for the cytosol, which includes the mitochondria. Here is the consumption for the ER and the Golgi apparatus. And in the nucleus, we didn't assume there was any consumption, but there was just a problem of diffusion. And the membrane part was then modeled by a simple permeability functor that basically reduced with the amount of cholesterol that was present. So it was an artificial modeling of the diffusion. So what we found was exactly that the oxygen, there was an oxygen gradient from the outside all the way through to the nucleus, and this gradient varied a lot on the consumption parameters, which are these V and K, which appear here in this equation, the Vmax and the Km. And we found that the lowest Km, or the highest Vmax, had the biggest impact in reducing oxygen. So just to give you an idea what that means, the low Km means the cell has become really efficient at dealing with oxygen. So, and most cancer cells have very low Kms. They have around 0.5%, and that's exactly the number we tested. We tested relatively low Kms. It means that the cell has extremely high efficiency at dealing with oxygen. So then we also took a study from the 1980s where they did ESR studies of oxygen uptake during the cell cycle, and they found that the 50% increase in cell cycle was observed between the G1 and the S phase, late S phase. So we took this number and we tried to understand if this cell cycle dependency could be modeled. So we did investigate that change, and we found that there is a significant change, in fact, in the oxygen level because of this higher consumption rate. And we found again that this was highly dependent on the Km, and the 0.5% has a big impact. Again, you see 0.5% here. Okay, so then we did some experiments in the lab to study, to see if the cell cycle was becoming hypoxic. Since we're predicting that in the model, we studied a variety of HIF1, HIF2, HIF3, and the PhD1s and PhD2 to see if there was really a change in the oxygen level, which we could really verify with HIF1 and HIF1A and 3A. And we then confirmed that a lot of this information of the HIF matched also the radiation response, that this increase of radiation response in the late ACE was related potentially with the cell becoming hypoxic during the late S. So just to finish this first part, cancer cells become hypoxic during the cell cycle. We showed that through experiments, but we initially predicted it with the model. Then we showed that the cell cycle, the cell itself, the oxygen enhancement ratio, depends heavily on the Km and the Vmax. And I just want to point out that we built this curve based on different cell lines. Most of them are healthy cell lines, and we used the numbers that they provided on the publication. And for example, CHO, which is not a cancer cell, but it's usually used in a lot of radiation studies, has extremely low Km, around 0.5. And there we found that the OER was around 3, 2.8 to 3, which matched a lot of the experiments. So the other thing I want to point out is FLASH does not depend on OER. I've just calculated the OER without the need of radiation. And basically what I've shown you is that the OER can be calculated as a ratio of oxygen in the nucleus relative to the ACM. Or we can do it like we've been doing for 50 years, is to use radiation. So we could use radiation as a nuclear oxygen probe or a microscope that tells you oxygen in the nucleus. But it's not something that depends on the radiation. We've used OER so many times to characterize radiation, but it shouldn't be. It should be used to characterize the cell and how the cell has the certain properties to resist radiation. So I've shown two ways, and I just want to bring this up, and I hope there will be discussion on this afterward. But we can use the radiation as an oxygen probe, right? It is a very powerful oxygen probe that tells you how much oxygen is in the nucleus. Okay. So that's the first part. I just wanted to give you an overview how important it is to separate oxygen from the cytosol and the extracellular matrix from the oxygen that's in the nucleus. I also wanted to differentiate the concept of OER, which is very specific to the cell, in fact, not the radiation. So I used this slide from Emmanuel Sifani about all these possible mechanisms on the FLASH. There are four main areas: the transient hypoxia, which is related to O2 depletion, organic radical recombination, the intertract, and the immune system. I'm not going to get into this one. I think there is one additional area that's missing that is related to redox chemistry. And I will discuss the Spitz paper from 2019, and the current talk will focus primarily on this redox chemistry and how that affects the FLASH. So I just want to remind you there are two potential pathways for oxygen damage. There's the one that was published in 2021, the Fauvedon, that has already been shown by Alejandro this morning. So you have the damage that occurs early. So you have either the direct or the indirect damage that's occurring. You produce all these radicals, and these radicals then can react with oxygen and produce peroxyls. So it is a situation where you have the damage occurring first, then the oxygen fixation, and you have these peroxyl radicals. So this is one mechanism I think is really a good way of presenting the data. I also want to present the one that we're working on in Heidelberg, which is slightly different, okay? And here in our case, the reaction occurs earlier. So oxygen reacts with ROS that is produced by the radiation, the solvate electron. And for me, this is an important molecule we should not forget about. And so the oxygen reacts with the solvate electron that's produced in large quantities by the radiation. And this ROS downstream will then eventually produce the peroxyl radicals. I don't provide a lot of the details on this pathway at the moment because we're in the process of submitting a patent on this whole process. But I do discuss how it works. So I also want to give credit to some experiments that were done more than 50 years ago, like this one, Ruth Roots, that showed the importance of the solvate electrons in these experiments. So you can block the solvate electrons by using nitrous oxide, N2O. And in this case, what they showed is that you get a much lower response. And so the survival was increased, of course, because you're blocking oxygen damage. Okay, so the solvate electron for me is one of the most important elements for the oxygen. I want to spend a second with that. So here's a schematic representation of the solvate electron. You have your solvate electron here in yellow, and it's usually attached to some solvent, which in this case is water. In reality, there can be a variety of solvents for the solvate electron. There can be the water version. Here you see water and alcohol, and you can also have different types of ammonia, amines, and you can also have even the hydrocarbonates can work as solvents. So there's a variety of solvents. In the case of the body, the most important is, of course, the water, which is this one here. So also, the solvate electron is not a free electron. It is a bound electron with a very low energy of around 3.4 electron volts. And it's really different from the free electron. That's what we measure when we do dosimetry. So dosimetry and solvate electrons are very different. But when we do radiotherapy, you assume that they're one-to-one, which they are not in FLASH, and I will show you. All right, so let's come down to the conventional. In conventional therapy, we give radiation, but we give it at a different dose rate than is standard. And in this case, there is very little intra-track and inter-track ROS interaction. So we produce all these ROS, and you see immediately that it's going to be very hard to them interact within each other. So in this case, because there's no inter-track reactions, you have the dominant of the reaction solvate electron with oxygen producing the ROS. Right? But in FLASH, you also have the inter-track effect. In this case, you have the competition of these two ROS elements which can produce the non-ROS. So here you have the solvate electron is going to be reacting with ROS, producing non-ROS effect. So notice that there's a solvate electron on both sides. The only difference is now I have oxygen on one side and the other one I have ROS. So this reaction is competing with the one that produces ROS. This one produces non-ROS. And if you do a simple calculation by hand, just to check, where does this 8 gray come from that we see so much in FLASH effects? We can really obtain it from this simple calculation. This is a hand calculation. You ask the question is, at what dose does this intertrack effect equal the oxygen effect? Right? It's a simple calculation in water. I don't even have to worry about the biology for now. And here's all the parameters. You can do it by hand. Anyone can do it. You have all the reactions. I put them here. You have to know the concentrations, which is relatively easy to obtain. And you obtain a simple reaction that looks like this. The oxygen in the nucleus is related to the dose. Right? And there's this constant here. Right? And this constant is obtained usually from the Wagner paper that tells you more or less what's the oxygen concentration at a specific temperature. Right? And you see, at 0.5% on the nucleus, I get 8 gray. And at 1%, I get 16 gray. So these are the numbers in which we have FLASH. Right? We have FLASH in this area of 8 to 16 gray. And I've just calculated in water, without including any biology, I've just calculated and obtained exactly the same numbers. That tells me that there is something related to radical-radical recombination. Okay, I have not discussed biology. Now here's the trick. The biology is the driver of the whole process, and it's driven by something called scavengers. Right? There's a variety of molecules that work as scavengers. There's like 8 to 10 molecules, and they all have different roles. I'm going to discuss them in a broad role so that you can see how it works. So a scavenger reaction usually has a curve that looks like this. There's a very rapid reaction rate, right? And then there's a saturation regime. And this rapid reaction rate is where it should always be working, of course. And this reaction rate is usually defined by a simple equation that looks like that. You have a Vmax, which is this maximum point of capacity for your scavenging system, and you have a ROS dependency on it. Right? And usually we have some called of a Km, or Michaelis constant, that defines more or less where the 50% point is. But the key parameter is the scavenging capacity on the left, right? And it is ultimately that is the driver of what's going on, in my opinion, for FLASH. Now, I want I've put the triangle here to explain to you the difference, right? So the scavenger will tell you which amount of Ross is left over to react with either oxygen or to react with the solvate electron in a radical-radical recombination. So the driver is not the Ross, but the scavengers, and how fast they work, okay? Now, let's look at this from a cancer perspective and a healthy tissue. Here is the Spitz paper, and the Spitz paper talks about reserve capacity, so I will discuss that in a minute. So let's see that I have a cancer which has a much higher Ross level than a healthy tissue, all right? If I have a higher Ross level, I will have a higher scavenging capacity in the cancer, of course. And in the Spitz paper, they talk about the reserve capacity. And this is correct, right? I agree completely with him. The reserve capacity for the healthy tissue is larger than the reserve capacity for the cancer. But there is a problem in this assumption. It assumes that the cell has not adapted to high Ross. So cancer cells will be constantly adapting to higher Ross. And if we do the adaptation, things change a little bit. So here's my healthy cell. Here's my cancer cell that is adapted to high Ross. 2 fractions, 4 times, 8 times. Now let's look again at this reserve capacity. It's in fact increased. So the reserve capacity has got stronger with Ross because the cell is adapting to the Ross. So you see clearly that as I increase the Ross from this cancer 1 to cancer 2, cancer 3, I have an increase of this baseline up here. Notice how much higher this baseline is compared to the blue one. It's significantly higher now. So the cancer cell has a much higher scavenging capacity. So because the scavenging capacity is much higher in the cancer, it's very unlikely that you will have this radical-radical recombination. It basically wipes out most of the radicals, and the few radicals that are left, some of them will react with oxygen, but very few. And there is where we start with the question, how does it occur differently in conventional versus FLASH? So let's look at the conventional radiation. So let's assume a simple pulse structure. I don't want to get into discussion of what is pulse structure or average dose or not. I put a simple dose rate structure here, pulse structure study, where I have in time and dose. And here is my scavenging system for my cancer. And the dose I deliver in a conventional is extremely small. I give very little dose. And you see that has no effect on the scavenging system. Yeah, right? The scavenging system is totally fine. That's the cancer. What about the healthy tissue? Well, the dose is small. So the Ross is small. Scavenging system is working perfectly. No stress. Everything's good. What about FLASH? In FLASH, now we have a high pulse, a high dose per pulse, sorry. And in this case, you have a giant step in Ross, right? You have a very high increase in Ross. Again, the scavenging system is working perfectly well. What about the healthy tissue? Well, now it's saturated. It can only deal with a small amount of Ross, and then it's fully saturated. Big problem now, you see? So now we have a differential immediately, right? The scavenging system for the healthy system is very weak. It cannot deal with high levels of Ross. The scavenging system for the cancer is very strong. It can deal with high levels of Ross. So let's quantify this. So here is my dose. Here's my biology. Here's my cancer, right? All this Ross is given below the threshold. The scavenging capacity is not saturated, it produces Ross. What about my healthy tissue? I have a different baseline. Now, if I do the same amount of Ross, now you see the difference, right? There's significant level of Ross smaller, right? So my first point is FLASH does not come for free. You will always produce some oxygen damage. The key is to control the dose rate, and I will show that how important the parameter dose rate is. And you see this is the amount of Ross, non-Ross, that doesn't produce damage. Let's look at the dose rate problem. So here is a dose rate of a very broad pulse, right? And you see a lot of it is producing Ross, and very little of it is producing non-Ross. Now let's shrink that pulse. I give more dose in a shorter time. I start to increase the amount of non-Ross and decrease Ross. And if I do this in a situation where I don't saturate at the cancer, you see the advantage of FLASH is dose rate. right? We have to look at FLASH not as a dose problem, but a dose rate problem. We have to increase this dose rate as much as possible without saturating the cancer response. And here you immediately see the impact of how physicists can work. We need to do dose rate escalation studies, both for healthy tissue and for cancer, and understand where the difference is for these two, right? So the healthy tissue, there's already been one initial study published in 2017 by Pierre Montegrul in the study for the brain, but I think we haven't done anything for the cancer. So the key difference is how to maximize FLASH effect. I was speaking to Alejandro and others. We want to really maximize this. Now, so one way we can do it as a physicist is maximize this non-ROS. We really want to increase this dose rate, get as close to the cancer as possible, but without saturating the cancer response. So that will be, in my opinion, the easiest way from a physics perspective. But there is also the biology part. Could we maybe potentially lower this threshold? Imagine that we could lower this thresholds to a point that we don't even need ultra-high dose rate. We could do everything with IMRT, 3D conformal, MPT, VMAT, you name it. Imagine that we could lower the threshold and we don't have to worry about which type of ultra-high dose rate we need. So that would be something that would be amazing. This requires, of course, some biological modulation of this threshold. Okay, so I wanted to finalize my talk with my final slide from Charles Limoni. So these are all the implausible ideas, and he's mentioned so many. I've added one additional one, which is the oxygen enhancement ratio. I hope today I've potentially convinced a bit of you that OER doesn't play a role in FLASH. I would almost say oxygen doesn't play a role, but I'm going to look forward to the debate tomorrow, so that'll be more fun just to sit back and listen to the debate. And I also want to raise the possible plausible mechanism. And Charles has already mentioned a lot of these in previous talks, but I want to add this additional part. We should look at the scavengers. The scavengers have a differential way of doing things, healthy versus cancer, and these can potentially affect how we do FLASH, in my opinion. I also have to thank all my postdocs, Jeanette, Francesca, Geraldine, Luca, and all the PhD students that have worked heavily in my group on this. I have several partners from Dresden that have done amazing work, Elke and Jörg. They've been absolutely fantastic in a lot of work we did. And I want to thank the people that sent me their slides, Emmanuel and Charles. And I think I can finish with a simple conclusion. Membrane cholesterol plays a role in oxygen diffusion. We have not looked at that since many years. I don't think we ever looked at it. And we should consider this. Then I also want to point out, in my opinion, there are two oxygen damage pathways. The one that was published recently by Fauvedon et al., we should look at that from an experimental perspective, and the one we're working on in Heidelberg that was already initially kind of suggested in the 70s and the 80s. Then the FLASH effect, in my opinion, depends on the differential response of the scavenging systems, healthy versus cancer. All right, sorry for the extra time, I apologize. I had a slight cough.
Thank you very much. Thank you very much. Is there any question from the audience?
Well, Joao, fantastic talk, by the way. A lot of questions. If you take your last hypothesis with the fact that you reduce the amount of ROS produced in the healthy tissue and then produce non-ROS, but then do not reach the cancer protection, what's going to happen if we're using a very high dose rate, like 10 to the 9th, 10 to the 13th, 10 to the 15th, as we are thinking about with some types of experiment, including VHEE?
So I think it's a very good question. I think the low dose rates we've been using for healthy tissue, say the 40 gray per second up to 100 gray per second, benefits healthy tissue. But I think as soon as we start going over 1,000 to 10,000 gray per second, it starts to slowly benefit the cancer. So I think we have to really see where is this window in the different regions we are treating. And I think we need to do a cancer study And it can be done in vitro for these high dose rates to see where the window, kind of where the threshold is for the cancer studies. We might try to do this in Heidelberg, but anyone that wants to work with us on these dose rate escalation studies, we're welcome to work with us. So I want to point out the in vivo studies can focus on healthy, in vitro can focus on cancer.
I have a second one, very quick,
Sure.
related to the aqueous electron, by the way. This one is pretty easy to test if you are trying to substitute the DNA of your cells with BRDUs, then you're gonna really enhance the sensitivity. Have you tested it? Have you tested it in conventional and flash conditions?
We have, yes.
And? So it does not work. We did as well.
Yeah. So I was thinking, almost to some degree, the flash effect could almost make a comparison to this effect produced by very high LT radiation density ionizing event, right? Produced by very heavy charge particles in this local environment. It produces a very large number density distributed electrons or others.
No, because chemically when you have high LET or high density, you convert everything into H2O2, so you don't have solvate electron. You'll notice with the LET, the solvate electron drops significantly fast.
Yeah.
So you don't need oxygen, in fact, for high LET for that reason.
I see.
Hi, Joao. Thanks for a very nice talk. A lot of innovative ideas, I would say. I have two questions. So first one is, what is, in your opinion, then the lack of a clear in vitro evidence for flash effect on tumor cells?
So there is evidence in the 60s and the 70s, right? The studies were done at extremely high dose rates of 10 to 6, 10 to 7. In the recent studies, we've done at low dose rate. So you'll see some of the papers at 40 gray per second. They are trying to reproduce the same dose rate as healthy tissue, and I am saying they shouldn't. They should take the in vitro data and go up in dose rate. Go up to 1,000 to 10,000 gray per second, and there you'll start to see again the flash effect clearly. So my main point is that what we're doing in the 60s and the 70s is very different from what we did now. So in the 60s and 70s we were here, and now we're doing this. So you see immediately that here I have only a small benefit, but in the 60s and 70s dose rate was so high, I have a huge benefit, and it should be clear. So my opinion would be the effect is there, it's just weaker because we're using lower dose rates. We should increase the dose rates for the in vitro cancer studies, and then you start to see a clear separation.
To which level increase? Since we use 10 to the 6 gray per second if we radiate.
It will be cell specific too, and we have to check the scavengers. So I think it's a good point. We'll have to try and see what happens. Why you at some dose rates you're not seeing it. I'm very curious.
Okay, thank you. And then the second question, exactly related to this type of graphs. So how would you explain, based on this saturation of the scavenging system, how would you explain the existence of the flash effect with both proton quasi-continuous beam and pulsed electron beam, where for the proton flash beam, okay, with 100 gray per second continuous beam, you're depositing less dose on a 1 microsecond level than in conventional electron beam. So conventional electron beam on a microsecond level gives you more dose than a continuous proton flash beam.
So I think we have a study for different LETs. One of my students has a poster. The key point is to look at solvate electron, how much solvate electrons we are producing in a certain time period. And you have to remember that protons have technically a higher LET than the electrons, so their production of solvate electron is different. So we'd have to look at the structure of this production and where it is. So maybe we could run a Monte Carlo simulation to check what is the solvate electron production in different types of particles and see how this produces the effect or not. I do have a student that's done that for different particles, starting at protons, but we didn't do electrons. We've done protons all the way up to neon, I believe. And so there's a poster showing the impact of the solvate electron as a function of LET. So maybe we could discuss that afterwards.
Yeah, I think it's a good idea to discuss this later because we are already late. So next question will be posed to Joao during the coffee break and when he has time to answer your further question. Sorry for that. And thank you once again.